we will hear leading experts telling us about their current research. I'm Christian Sterz, Science Counselor to the German Embassy to Brazil, and I'm happy to guide you through this event. This event is organized by the Brazilian Ministry of Science, Technology and Innovation, MCTI, the German Embassy to Brazil, the National Council for Scientific and Technological Development, CNPq, and the German Center for Research and Innovation in Sao Paulo. And I would like to thank all of these organizations for their support to make this event happen, as well as the speakers who took their time despite busy schedules to contribute to the exchange, and last but not least to our audience to take part in this event. Now I would like to pass over to the speakers for the opening remarks, and I invite Mr. Marcelo Morales, Secretary for Research and Scientific Training from MCTI, to speak to us. Mr. Morales, it's a great honor and pleasure to have you with us today. The floor is yours.
Good morning, Christian. Thank you very much. I would like to thank you, the Jochen, the Director of DAAD Brazil, Dr. Evaldo Vilela, President of the National Council for Scientific and Technological Development, our Científico, and the Ambassador, Heiko, Thoms, for the opportunity to talk with you again. And since technology and innovation are some of the most relevant areas in the German-Brazil relationship, I would like to reinforce Germany is the priority country for cooperation with the Ministry of Science, Technology, and Innovation. And since 1996, MCTI, BMBF, and their partners have periodically met to discuss science, technology, and innovation cooperation in the Joint Commission on Science, Technology, and Innovation. About 1 year ago, it held the 29th meeting in the joint German-Brazilian Committee for Cooperation in Science, Technology, and Innovation, and with the working group on priorities in life science and health research, identify lots of topics of common interest. In this context, this webinar series was jointly planned by MCTI, CNPq, and German Embassy in Brasilia. With these meetings, we hope to broaden and strengthen even more our joint action. Today, we will focus on lessons learned from the COVID-19 pandemic for the next emerging viruses. In order to develop the discussion on the Brazilian side, we invite some of the researchers from MCTI's Committee of Specialists that we call Rede Virus MCTI. And since the beginning of 2020, the Ministry of Science, Technology, and Innovation of Brazil has mobilized research units, Institute of Science, Technology, and Innovation, and laboratory spread through Brazil in response of new coronavirus emergency. These institutions are working together and focusing on clinical trials and projects that are increasing our understanding of the efficacy and effectiveness of vaccine therapeutics and clinical approach to COVID-19. The kickoff of this initiative was happening in February, and I would like to emphasize it was February 10, 2020, when the MCTI promoted the meeting, counted on the participation of renewed experts and representatives of the Ministry of Health, scientific entities in the National Institute of Science and Technology, who discussed the contribution of science to the confrontation emerging viruses, especially coronavirus and influenza. As a result of this meeting, it was concluded that there was a need of formation of committee of specialists to be instituted with the scope of MCTI, whose primary function will be to contribute to the articulation and integration of research, development, and innovation related to emerging viruses. This committee helped the MCTI to decide the strategy for research and development to combat COVID-19. We highlight some of the strategies. It was diagnosis tests that we developed in Brazil, drug repositioning in medicine, preclinical evaluation of new molecules that we have already in Anvisa, clinical trials, more than 35 clinical trials only here fostered by the MCTI, but the entire country, more than 200 clinical trials. Support that we increase the level of laboratories from level of security 2 to 3 in 18 institutions. And we studied the impacts, economic and the social impacts of COVID-19. And we have in this subgroup of Rede virus, more than 40 publications about the impact. Sequencing, we are responsible at the MCTI, Rede virus MCTI, more than 65% of the sequencing of the virus in the entire country. So many effort from the Brazilian community that I am really very proud about our scientists, and we can join together with Germany this effort to avoid new emergencies and viruses that we know that we are going to have new pandemics in the future, and we have to be prepared. And it's very nice to count on Germany in this partnership. Thank you very much.
Christian, seu microfone está desligado.
Muito obrigado. Many thanks, Mr. Morales, for your important speech. It shows the high level of research already done in Brazil and its efficient organizational structure. And it seems to me an excellent starting point for bilateral exchange and cooperation. And an excellent starting point to find German partners for bilateral cooperation is, of course, the German Center for Research and Innovation in Sao Paulo. At this point, I welcome Jochen Hellmann, President of the representation of the German Academic Exchange Service, DAAD, in Rio, and in this function also responsible for the German Center for Research and Innovation in Sao Paulo, to our webinar. Dear Jochen, we are looking forward to your contribution.
Thank you. Thank you very much, Christian. Dear Marcelo Morales, Secretary for Research at MCTI, dear President of CMPK, Evaldo Vilela, and dear Ambassador Heiko Thoms, dear participants and everybody who has contributed to this event, dear speakers, of course. As Christian mentioned, on behalf of DWIH, German Center for Research and Innovation, and of DAAD, the German Academic Exchange Service, here speaking from Rio de Janeiro today, I would like to extend a warm welcome to all participants and all contributors to today's event. Lessons learned is the subtitle, lessons learned from the COVID pandemic, of course. I think the relevance of this topic is quite obvious, and nevertheless, I do even if I do not mention the necessity of speaking about this subject, I would like to underline how great the damage caused by the pandemic is, and how many victims there are in many countries of the world, and how great the social and economic damage is, even for those who have been spared itself by the virus up to now. It is clear that the focus of all of us must therefore be to ask, how can we prevent such a disaster from happening again at the next opportunity? What do we need to do better to be better prepared the next time? What does science exactly know, and what does it still need to research in order to be able to advise decision makers even better, and to provide the social and medical answers to the next challenges? The DWIH sees, of course, its task precisely in supporting and promoting events like today's. Representatives of German and Brazilian science exchange their findings on the burning issues we are aware of, learn from each other, share their ideas, also their data, and perhaps their students, and cooperate with each other. Because no country can face the challenges alone. It can only be done with international cooperation. In DAAD, we are absolutely convinced of that, and I know that the German government will always promote this idea. International cooperation in science is the point. The virus has no regard for national borders, and cannot be stopped, not even by oceans, from spreading all over the world. And so we should also keep in the earth as a whole in our mind, and learn from each other, so that we can say, after this crisis, honestly, and perhaps even a little bit proudly, lessons learned, the title of today's event, and was a subtitle. Now I wanted to be brief, not to take too much time from those who have scientifically more to say about the topic than myself. But now I wish all listeners, all participants, and all those who are with us today an interesting and insightful seminar, and thank again everyone who made it possible. Christian, please.
Thank you very much, Jochen, for your very optimistic speech. Surely the German Center has a lot to offer for future health cooperation with Brazil. I believe cooperation based on institution is key to a sustainable cooperation base. Which brings me to our next speaker, Mr. Evaldo Vilela, President of the CNPq. Surely the CNPq is one of the core institutions in Brazil that have to be included in an institutional cooperation, not only if you're seeking EU funding for joint projects. Evaldo, great to have you with us today. Please share your ideas with us.
Thank you very much indeed. Good morning to everyone in Brazil, and good afternoon for the Germans in Germany. I would like to greet Dr. Marcelo Morales, Secretary for Research and Scientific Training from the Ministry of Science, Technology and Innovation, Dr. Jochen Hellmann, Director of the German Center for Research and Innovation and German Academic Exchange Service, DAAD, in Brazil, and also our Ambassador of Germany in Brazil, Dr. Heiko Thoms, and all scientists and stakeholders that are taking part in this webinar. I wish you all a good day. The scientific and technological cooperation between Brazil and Germany has been lasting and strongly supported by academic exchange and joint research projects. The two countries meet periodically to discuss cooperation in science and technology in the Joint Science, Technology and Innovation Commission. It's important to remind that such cooperation has been developed in a number of areas such as bioeconomy, health, IT, ocean, advanced materials, and environment. In particular, it's important to mention that the Observatory of Tall Tower in Amazon is an initiative generated under the cooperation between Brazil and Germany. We must continue to cooperate. Health is one of the areas that has demonstrated a special condition to be developed as a must under the umbrella of the mentioned cooperation, and that is why we are here today to discuss the lessons learned from the COVID-19 pandemic developments in science and technology. Since the end of 2019 in the world and the beginning of the 2020 in Brazil, COVID has affected societies with an unknown disease. Governments moved in search of knowledge capable of fighting the novel coronavirus outbreak. It's an outbreak never seen in recent history that has generated disastrous consequences and has threatened global security health. Scientists around the world spared no efforts to bring possible solutions to fight the pandemic because it showed the world once more that science has to be done together. On February 10, 2020 in Brazil, experts and representatives of the Ministry of Science, Technology and Innovation, Ministry of Health, scientific entities, and national institutes of science and technologies that we call INCTs, among others, gathered to discuss the contribution of science to the confrontation emergence viruses. As a research, the Minister of Science, Technology and Innovation has instituted and named the hedge virus, the network virus, as Dr. Marcelo has stressed, with the aim of integrating collaborative initiatives to combat emergency viruses with a special focus to COVID due to the pandemic. So altogether, Brazilian institutions have worked on the diagnosis, treatments, vaccines, and the production of knowledge about the virus. The National Council for Scientific and Technological Development, the CNPq, has an important role in promoting science and the training of human resources for research in Brazil. Has acted in various activities together with the Brazilian Ministry of Science and Technology in combating the coronavirus and its consequences. Among them, I will cite some examples. We had a public call, CNPq, Minister of Science and Technology, and BRICS in 2020 to support joint research, development, and innovation projects in the following areas related to COVID. First of all, research and development of new technologies and tools for diagnostics. Research and development of vaccines and drugs, including the reposition of available drugs. genetic sequences of the virus and the studies of epidemiology and mathematical modeling of the pandemic. And also research-oriented artificial intelligence, high-performance computing of new drugs, treatments, clinical trials, facilities, etc. And also epidemiological studies and clinical trials to access SARS-CoV-2, overlap and other comorbidities, in particular tuberculosis. The resources invested in the action totaled more than $1 million to support 12 projects. Among the BRICS, Brazil was the most represented country in the above-mentioned call. Some other actions. We had a public call, Científico, Minister of Science and Technology and Innovation, in 2020, called HIGH Program, which is about a research in business incubation, when we approved 3 proposals associated with projects aiming at combating, preventing, and confronting COVID-19. And also some supplementation of an ongoing projects are the INCT projects on public communication of science and technology, scientific dissemination about COVID, INCT sustainable treatment stations, development of project COVID storage monitoring network, projects of the Information Technology Center, Renato Asher, which involved nanotechnologies to control the COVID pandemic. Project of the National Center for Research in Energy and Materials, CENPEN, and also that involves nanotechnologies. Actions to support the health areas in maintenance of medical and hospital equipment, inclusive through additive manufacturing. Pragmatic trials, clinical trials to evaluate the effect of masks with chitosan nanoparticles for filtration, potentiation, and biocidal effect induction against coronavirus. These are examples of actions implemented by Científico and the Minister of Science and Technology in response to the pandemic. Certainly, many lessons have been learned and others are in process, so that in partnership, we can better prepare for the next emergence viruses that will challenge us in our future. I wish you all a morning of a good discussions. Thank you very much.
Thank you very much, Evaldo, for your speech and your showing your deep involvement in current research projects. In fact, the idea of organizing this webinar emerged in an exchange of ideas between CNPq and the embassy about possibilities for future bilateral cooperation. And we took a lot of advantage of your scientific competence. As our final speaker of opening remarks, I heartily welcome His Excellency Mr. Ambassador Heiko Thoms to our webinar. I remember well how the need for more bilateral cooperation in health on the backdrop of the pandemic was raised by yourself in a meeting with Minister Pontes and Secretário Morales already early in 2021. Now we witness today's webinar as part of this bilateral engagement. Mr. Thoms, we're looking forward to your opening remarks. The floor is yours.
Thank you very much. Well, dear Secretary Marcelo Morales, dear President Evaldo Vilela, dear Mr. Hellmann, it's great to see you all again, if only online, and I very much hope that at some point when we continue this process, we will be able to hold a real seminar also in person, bringing together Germans and Brazilians. Ladies and gentlemen, bom dia a todos y todas, guten Tag. For many people, the COVID-19 pandemic has been a defining time of their lives. It has changed the world in a lasting and substantial way, and it continues to usher in yet further changes. It is my great pleasure to welcome you on behalf of the German Embassy to this event on lessons learned from the COVID-19 pandemic for the next emerging viruses. And while I'm here, I would like to take this opportunity to express my sincere thanks to everyone who has helped organize this event. And special thanks go to today's speakers who have generously agreed to share their insight despite their busy agendas. Global challenges can only be surmounted through international cooperation. That is something the COVID-19 pandemic has again made painfully clear. It was international cooperation between scientific experts and top researchers that made it possible to develop vaccines in record time and to reach a better understanding of the cause of the pandemic. The first mRNA vaccines in the history of the world have been approved. This new type of vaccine represents a technological advance, a breakthrough, and a great scientific success that seems, as far as we can judge today, to be living up to its promise. We must continue with this important work in research and development. It is already evident that we will not be able to eradicate the SARS-CoV-2 virus. It continues to mutate and requires constant monitoring. As a result, new vaccines that target the new variants will constantly have to be developed. Furthermore, we know that this will not be the last pandemic. Countless viruses unknown to humankind are hidden in the melting ice. And the wildlife of the planet continues to play unwitting host to a stream of new viruses. The latter are particularly relevant due to the considerable contact and increasing contact between wildlife and humans. Today's event is intended to launch greater exchange between experts, also on questions of preparedness for future pandemics. We want to learn from our experiences during the COVID-19 pandemic and draw on these lessons to protect ourselves from the pandemics of the future. Germany and Brazil are in an excellent position to cooperate on work to combat pandemics, both having the necessary expertise, experience, and framework conditions. Both countries have a large number of outstanding scientists and research institutes. Brazil has already experienced many epidemics, and with its network virus MCTI committee, has an excellent organizational framework to deal with them. In addition, Brazil is home to a huge diversity of wildlife and is an indispensable partner for research on zoonotic diseases. So we know that international research cooperation is called for, but we also know that international research cooperation requires trust and a stable basis on which to build. These preconditions normally emerge through personal contacts and initial joint projects. It is thus by no means surprising that the cooperative development of vaccines has, as a rule, built on existing collaborations. Or, in the words of Henry Ford, coming together is a beginning, keeping together is progress, working together is success. We want to do all we can to make our cooperation a success. Accordingly, today's webinar should not remain a one-off event, but should be the first of a whole series of webinars, which will ideally give rise to enduring friendships, joint projects, and exchange at institutional level. On this note, I wish us all a successful event. Thank you very much for your kind attention.
Many thanks, Mr. Thoms, for your speech. I know this Brazilian-German cooperation in health is very important to you, which reflects well in your own engagement and support. Many thanks for this. Now I would like to come to our sessions. In all panels, we have an introductory presentation from Germany and one from Brazil. Then our scientific audience will have the possibility to enter into a direct exchange with the speakers, and you're invited to comment on the subject and to ask questions to the speakers. Our first session is on genomic surveillance, and I have the honor to introduce to you Professor Fernanda Spilki and Professor Drexler. Professor Fernanda Spilkey is Dean of Research, Postgraduate Studies and Extension at the University of Vilela and member of the MCTI Virus Network and the coordinator of the MCTI FINEPI Corona Omnic Network. He has a master in veterinary science in the area of animal biology and a PhD in genetics and molecular biology. You work on projects in the area of animal, human, and environmental biology. Mr. Spilkey, many thanks for joining us. I guess nobody else here could better describe the current pandemic situation in Brazil as you can. What are your experiences and which challenges do you encounter in your work? Mr. Spilkey, the floor is yours.
Hi, thank you, Christian. It's always a pleasure to talk with you, and I would like to send a good day and good afternoon to our colleagues and also for the authorities that are here that presented about the status of the research in Brazil and about the collaboration. I would like to thank also for all the authorities to how they praised our work, because it's good to hear from you that scientists have helped in the combat of the pandemics. Thank you very much. I would like to ask whether I could put my first slide, please. I think that this is the last one.
Can you share your screen, probably? That
Yeah,
would be the easiest way.
Maybe I'll try to share my screen, maybe, because I'm not allowed to share this.
Okay. Yeah, can you help?
I am sharing your presentation. Can't you see it?
Yes, yeah, but I unfortunately, this is the last slide.
Oh, I'm sorry.
Thank you.
Sorry about that.
You are showing some spoilers of my presentation right now. Okay.
I apologize. There we go. Thank you.
Okay, thank you. Oh, so this is an honor to be here. I will present briefly some of the works of the efforts that we did in terms of genomic surveillance here in Brazil. I'm representing, as the coordinator of the Corona Omnica Network, more than 100 scientists that are working every day routinely with the characterization of the virus that is circulating in Brazil from the very beginning of the pandemics in the country up to now, and we will continue these efforts, I promise. So, next slide. At the very beginning of the crisis, when the problem was constrained in the city of Wuhan, we were joined as an invitation of the Secretary Marcelo Morales in Brasilia to talk about what could be the priority targets in terms of research to deal with the pandemics in the case that the virus became a pandemic and the virus came to Brazil. And one of the priorities that was discussed by the experts that were there was about the necessity to have a very organized genomic surveillance system to deal with the circulation of the virus in Brazil. So next slide. We formed this Corona Omnica BR. This is sponsored by FINEPI and the Científico, and this is an initiative of the Virus MCTI. We were at the very beginning 12 institutions, from universities to centers of the government, federal government itself, to do sequencing and to also work with other omics that could be used to understand the disease and not only the circulation, how the virus is evolving in Brazil, but also about the response of the patients in terms of exomics and transcriptomics. And the fact is that nowadays we are more than 20 institutions, and we have not only this mission to do genomic surveillance and other studies, but also we have this mission of build capacity to form new researchers that could deal with this sort of confrontation with, especially in what regards to genomic surveillance and epidemiological studies linked to genomics. Next slide. We have this website that is available as it's very easy to find this in any of the searching tools of the internet. And there, next slide, we not only publish our academic studies, these papers that are being published in peer-reviewed journals, but we have also reports of the situation that we published at least once per month. Next slide. And we, inside this website, which is based on our National Laboratory for Scientific Computation, the LNCC, in Petrópolis, Rio de Janeiro, we have all the information regarding these sequences that are sequenced in our project. But we do not only put these sequences at the GSAID and other international databases. We have also access and we have deposit of all the clinical data and the raw readings of the sequencing there at this great web server. So this will be something that will be there for the future to study all other possible variations that have occurred during time in the virus and the partial species that are there, and the relationship with other clinical data. Next slide. And we have also automated tools to deal with variant analysis, and these allow us to really share in real time any possible new variants that come and that could appear, not only being imported, but also those that are autochthonous to Brazil. We could share this information with other authorities. Next slide. And we do this through a special channel of communication between the Ministry of Science and Technology and Innovation and the Brazilian Ministry of Health. We are in close contact in terms of communication, and we also published all this new information in the website of the Ministry of Science and Technology and in the website of Rede Virus. Next slide. Today we have more than 70,000 Brazilian sequences, SARS-CoV-2 sequences in the basis of GSAID, and I will say to you that most of these sequences are linked to or were done by laboratories from our network, and we jumped from the last year of an average of 400 sequences per month to today something like 4,000 sequences per month, and we are trying to reinforce these with other initiatives and the continuation of this network. Next slide. And for some of the results, we did the first description of what was after called the Zika variant under investigation, which is also called P2, at the very beginning of the formation of this new variant in Rio de Janeiro. Next slide. One of our groups at the Institute of Tropical Medicine in São Paulo was one of the first that described the P1, the gamma variant of concern, in Manaus. Next slide. And we have also mapped the dissemination, the dispersion of these new variants throughout the country, like this work that we have done here in Rio Grande do Sul at the very beginning of the P2 spread in Brazil. Next slide. Co-infections between different lineages. Next slide. And many older lineages that appeared between the end of 2020 and 2021, like the variant NP13L and variants NE9, N10, and many others. Next slide. We have also described a new variant very close in terms of genetic background to those variants that come from Delta, but of course this variant was a descendant from a different lineage, and this variant was named P4. It happened in the middle of the first semester in São Paulo. Next slide. And it almost occupied the space of variant Gamma at this very time, but Gamma, at the end, Gamma won this battle as it won in every part of Brazil. The battle, even against the Delta variant, it was a huge fight that Delta have a lot of trouble in to get the space that Gamma had. Next slide. We have described a lot of works with reinfections. Next slide. And the possibility of sharing of the virus by reinfected people here in Brazil, and this was important especially to talk with people in the front line, with healthcare workers about this possibility. Next slide. And we have also conducted national studies in terms of the rate of mutational events that we have here in Brazil. Next slide. Our mutational rates were compared only to those that were found in India and in Africa do Sul, and Southern Africa, sorry. So it is remarkable that Brazil have brought the space for a lot of variants to become reality here. So at the end of this process, we had this Gamma variant. Next slide. That was the variant that have dominated the whole scenario in the first semester of this year in many of the Brazilian regions. Next slide. And it is also important to share with you the knowledge that we have to reinforce the surveillance along our borders, and we are doing this right now in partnership with our Argentinian colleagues. And it is remarkable that we have found, even during the Gamma outbreak, we have found some new variants that have spread between the borders, and this is something that we will reinforce in the near future, and it is an initiative of the Minister of Science as well. Next slide. And today what we are having in Brazil, and I remember that I discussed this with Christian a few weeks ago. It is about the protagonism of the Delta variant nowadays in Brazil. Not only Delta, but some of the branches that come from Delta, especially nowadays, the AY43 branch and the AY33 branch. These are the viruses that are dominating the landscape nowadays. And this is a result that comes from our network, showing that the same genotypes that are nowadays the common genotypes in Europe are also the common genotypes dominating the scenario in Brazil. Next slide. And as a top take-home message, deal with genomic surveillance does not end at the sequencing of the virus. Of course, we have to study all the biological, the phenotypical factors that come from any new variant, and we are doing this. Next slide. We have done studies about the neutralization of these new SARS-CoV-2 variants that occur in Brazil, not only with gamma, but we have other studies being done with other variants that are here about the immune response from people vaccinated with different vaccines available. Next slide. And also we are describing when it happens about the outbreaks that could occur even in vaccinated individuals. We are searching for any new mutations that could occur in this public. Next slide. So this is it for this morning. Of course, I'm open for questions after the presentation of our colleague, Dr. Drexler. So thank you very much for this opportunity, and I'm here for any questions that could come from the audience. Thank you very much.
Many thanks to you, Mr. Speaky, for this very important input. It seems to me as if your findings are crucial for all of the world, yet not all of the world is contributing to your work yet. Our next speaker in this panel is Professor Drexler. Jan Felix Drexler is Professor of Virology at the Charité in Berlin, where he has the Virus Epidemiological Working Group. His work and research is focused on novel viruses from humans and animal reservoirs and the possible virus transmission and spread from animals to humans. In the course of his research, for instance, Professor Drexler traced back the mumps virus to bats and demonstrated that they also can carry hepatitis B viruses. He is also a recognized expert on Zika virus. Mr. Drexler, many thanks for joining us here despite being in Africa at the moment. And you have a long-standing cooperation with Brazilian experts. Did you ever meet Mr. Speaky before? Please tell us about your research findings and your cooperation with Brazil.
Thank you very much, Mr. Sters. Thanks everybody at the embassy for organizing, at the Brazilian authorities, the CNPq, the MCTI. This is a really important and very useful event. It's rare that we have those meetings of scientific and public stakeholders, and we need more of those, obviously. In reply to your question, Fernando and I met, we met recently, but we have huge synergy. We have just met again virtually at the Brazilian Congress of Virology, and he was president of the Brazilian Society of Virology for a few years, and so it's a real pleasure to be united once again, virtually, unfortunately, with Fernando. I also note my friend Durigon is from Sao Paulo, is also on the talk. Hi, Durigon, long time no see. We have a long-standing history in Brazil. I worked for 8 years in Salvador, Bahia. I speak Portuguese. I have 3 kids who are Portuguese. No, sorry, who are Brazilian and German. My wife is Brazilian, so it's really my second home. And I'm very happy, grateful for this invitation. Having said this, let me get straight into matters. We are a bit late. I hope the internet will work. As you mentioned, I'm in Western Africa and internet has been somehow unstable today. This is about the experience, and my talk is a bit different from Fernando's because I will show some data, but much less so. I'm really trying to go more for lessons learned. Let's go for this. Next slide, please. this is just to highlight again that if we talk about COVID-19 and Latin America, we are looking at the global hotspot of the pandemic, Brazil being one of the countries hardest hit, as in this overview where a darker tone of blue indicates a higher number of cases, and Latin America is really suffering on the individual, the population, and the economic level. Next slide. We have many projects in the region. This is every country that is colored in yellow is a country where we have bilateral research projects, and you can see that most of Southern and parts of Central America are pretty yellow. I would like to highlight the Dialogue of the Pandemic, which is a project I coordinate, which is funded by the German Ministry for Foreign Affairs, and where we try to do exactly what you are doing today, to talk to one another and learn from one another. And we are organizing symposia where we try to establish transnational cooperation between our Latin American colleagues and German scientists. We have many other projects beyond that one in the region, funded by different German federal ministries, including that of research, that of economic cooperation and development, and we also have EU-funded projects. And we're looking to future initiatives to establish and to strengthen transnational infectious disease research in the region, hopefully funded by the European Union or the Ministry of Foreign Affairs. We shall see. Next slide. The diagnosis, and we are in a genomic surveillance session. I'm extrapolating this genomic surveillance to just detect viral nucleic acid by molecular methods, such as PCR. You can see here in this graph the increase of tests, of PCR tests, done over time in different regions of the planet. And as you see, Latin America is second only to Africa in a relatively lower number of tests that are done in the subcontinent, and a relatively less intense increase. Compare this to Europe or Northern America. So we are really looking at a region globally that has, for many different reasons, I will come to this, has encountered challenges towards the molecular diagnosis of COVID-19. Next slide. And the relationship between diagnostic capacity is not easily understood, and a relationship between diagnostic capacity and well-being. This is three plots that align, that correlate the number of PCR tests on the x-axis with different socioeconomic indicators on the y-axis: the Human Development Index, governance, and urbanization. So this is different regions. Latin America is shown here in well, call it pink. And all you have to see here is that all those lines are pretty linear. So it means that, maybe not surprisingly, the richer a country is, the more tests it can pay. But there's a lot of hidden data in this. I will not go into detail, and you please do not try to decipher these small data points. Governance plays a central role, and urbanization, and on the contrary, the decentralization of diagnostics to rural regions remain a constant challenge across all of the tropics, including Latin America. Next slide. Genomic surveillance, what we are discussing in this session, takes time and resources. This is a really expensive task. Fernando has shown a slide with his funders. So this is, I would like to congratulate especially the Brazilian funders for going for this endeavor together with our Brazilian colleagues. This is really, it's incredibly important, and it's incredibly expensive. So you are doing a great job in funding this. You see that Delta has gone global on the on the planet, highlighting the value of genomic surveillance. You see the increase of Delta here in light blue in this plot from NextTrain. In Brazil, Fernando has mentioned this, you really need to do these detection and sequencing studies to understand what is going on in your population. And viruses do not respect borders. Fernando has mentioned this, and I completely agree. We need to engage in this transnationally. Next slide. We have also, I am now in Western Africa, in Benin, and I would like to highlight that together with the head of the reference laboratory in Benin, Henri Adulton, we have been able to provide the first epidemiological study of the Delta dissemination in a peer-reviewed journal. It should come out pretty soon. It's not available, unfortunately, yet as PDF because the journal is taking incredibly long. But you see here in this highlighted in red how Delta took over in Western Africa in a matter of 6 weeks, from 3% detections to 60% in little more than a month, highlighting how much more transmissible this lineage really is. And that's all the more worrying if we have to consider the poor vaccination status in many of those countries. Next slide. I would like to highlight that in our work with Brazil, we have always engaged and tried to contribute to excellent diagnostics. This is data from an external quality assurance or assessment, EQA, that we have done on the capacity of Brazilian expert labs to detect Zika virus. Brazil was hard hit by Zika. It's the country globally hardest hit by the Zika outbreak in 2014 to 2016, associated, as you all know, with microcephaly in newborns, dramatically so. And we were able to show, together with our Brazilian colleagues, that many labs performed excellent. These are the gray bars that go to the top. Some laboratories had problems, that are the bars in cyan that go down in this plot. That includes false positive results. So that means that you have a sample, say from a pregnant woman or from a neonate with problems, and you provide a diagnosis to this individual, yes, you are Zika infected, and you give an explanation for something which is potentially a congenital infection or a malformation. That is not easily done, and you need reliable diagnostics to support public health considerations. Next slide. Because dramatically so, we are still debating what is the actual risk of microcephaly upon maternal Zika virus infection during pregnancy. I won't go into details, but the risk estimates for that to happen if the mother is bitten by a mosquito during pregnancy and is infected with the virus, the Zika virus, so that it can pass on to the baby, to the fetus. We are really not fully understanding this still, and we need diagnostics to reliably understand the data from our cohort studies. I would also like to highlight that abortion is illegal in most parts of Latin America, not all, and not all treat that difficult topic equally of the of the Latin American governments. But we know that abortion requests due to suspected Zika infection or generally during the Zika epidemic have increased dramatically by over 100% in pretty much all of Latin American countries. So again, I would just like to highlight, do not interpret laboratory molecular surveillance as something that is just for a weird scientist, a weird virologist in his little lab. That's not the case. There is a direct translation to public health, and we need to fund those initiatives urgently so that we can progress as societies and on the whole. Next slide. I would also like to highlight, I'm showing now, we have conducted the first Zika virus, the first SARS-CoV-2 EQA. Remember, external quality assurance, you send blinded samples to the labs and see how they do. And you inform them if they don't perform well. And we've done the same in Europe for the European reference laboratories from the European Center for Disease Control and Prevention. And this is the results, next slide, of that analysis. It was published a few months ago. And again, I've highlighted false positives here and false negatives. Again, that is just wrong test results. I'm showing this because I do not want any audience to interpret my Zika test slide as if that was a particularly Brazilian problem. This is a global challenge to ensure adequate molecular diagnostics and genomic surveillance. So this is why I'm showing this slide. These are the European reference labs from 60 European countries. This is the top level of pretty much all of Western Europe and large parts of Eastern Europe. So those are the best laboratories, public laboratories we have in Europe, and they have wrong test results because this is what happens. It's not easy to run those tests, especially if you have limited resources and a high demand for tests. Next slide. And that brings me to my last 4 to 5 slides. I will be very quick on those. What is challenging? What are the lessons learned from COVID-19? How can we do better in the next pandemic? First, affordability and access to reagents is a challenge in tropical regions. It's hard to buy expensive reagents in local markets. Latin America and Brazil, all my colleagues now in the talk are nodding their heads because they have it from a personal experience, and I share that experience. It's expensive, it's hard to import, it takes months. This is not compatible with pandemic response. Next slide. The second constraint, the constant lack of decentralization of diagnostics in Latin America and all of the tropics. We mostly do well in the big capital regions, Rio, Sao Paulo, but we need to do well in the provinces of a country. This is data we just got accepted. The paper will come out in, I hope, a few days, from Peru, a country with almost the highest mortality per capita from COVID-19, hard hit. And this map shows a rural region of northern Peru, where on the left side you have 0 prevalence that we determined together with our Peruvian colleagues, and on the right side you have reported cases, incidents. All you have to do here is to follow me that the colors don't match between the two maps, and they should match. It shows you how you have a disconnect between reported and notified cases and real infections determined by antibody status. So this is all the more worrying in rural regions where access to healthcare posts is difficult. People don't have money to go to these places. The labs are under-equipped. And if you are a day laborer or working for self-sustenance in the countryside, you cannot afford mandatory quarantine. You cannot afford to be put in a hospital for 14 days because your family will not have food to eat. You need to go out and work. So probably you avoid getting a test. So this is a complex setting I'm trying to summarize in this simple slide of socioeconomic, cultural, and economic factors, all together challenging rural detection of cases. Next slide. Human resources is another constraint. We lack trained health professionals. We have a constant problem of migration of trained personnel from the tropics, including Brazil, including Latin America, where those that your systems have funded, have graduate studies, postgraduate studies, then leave the country to work, say in Germany, say in the United States. So this brain drain is a constant problem in Latin America, and the answer to this is we need to ensure adequate settings, adequate funding for science, so that we can keep the best brains of those countries that know the system, that know the countries in their settings. Of course, I have many Brazilian students. I'm not saying they shouldn't come to my lab, but we have to be careful to avoid a brain drain, how it was established in the 80s or in the old days. This must not happen, because we have to work together. And COVID-19 is a pandemic. If we don't work together, we will suffer and we will fail together. Next slide. Governability is a big issue, and I don't want to talk politics because I'm a scientist and I don't have to talk politics, but I would like to highlight how complex in Latin America and in Brazil the coordination between the central government and local authorities, say on the state level, say on the municipal level, can be. You need the public stakeholders to join forces across political parties, and we have to overcome left-right dichotomies, how they are established in Latin America, and unfortunately that are hard to be overcome. I think this is a lesson from the pandemic. We have to work together. This is not a political game. This is health of everybody that is on the planet, and if we are stuck in this left-right thinking, we will again delay our response. And if there is one big lesson from the pandemic, then is this: we must never again be as unprepared as we were with COVID-19, and this means we have to engage together. Next slide. Last slide. I thank all my group, which are shown here, and all the funders. Thank you.
Many thanks, Mr. Drexler, for pointing out the challenges of diagnosis and the possible risks going along with this. Testing and sequencing and public opinion is believed to happen in due manner. Yet there still seems to be many blind spots. With this, I'd like to open the floor for discussion for our audience, and we already have one question from the YouTube channel from Alex Cordas. How to use epidemiological data on COVID and trust if these data were purposely mixed with other respiratory diseases? Who would like to answer first? Fernando?
This is exactly one of the challenges that we have right now. I think that one of the things that have to be part in the aftermath of this pandemic is that we could maintain this network. But fortunately, we have fewer cases in Brazil today. But this network is formed, and this could serve for exactly for this kind of effort to follow the evolution and the distribution of different viruses, and many of these are endemic in Brazil, like this one cited by Dr. Drexler. We have Zika, dengue, and many respiratory diseases. Of course, we need to do much more effort, especially for influenza, and I think that SARS-CoV-2 may be in a lower frequency in the next years, but we'll have to look very deeply for his evolution in this post-vaccination world. So I think that one of the things that have to be done as a legacy is to reinforce these and other networks for study of the virus. One of the difficulties that we have is that we have the expertise. We have many laboratories formed before the pandemics, but these laboratories had some funding that comes from older pandemics, like the Zika pandemic in 2015 and 2016. And that takes a lot of time to have all these projects approved and have all the money that is necessary to run the routine efforts when you have to start from the very beginning, like we had. So this is why our efforts and our results are so good this year, because the money could come to our labs. That was the time needed to have this. So one of the things that we have reinforced, and that was excellently pointed by Drexler, is to have a more continued funding for this kind of efforts. But of course, we will need to do much better surveillance, not only for SARS-CoV-2, but also for arboviruses, respiratory diseases, and I would like to put in the room also enteric diseases, which are very important in the case of Latin America as a source of disease, namely gastroenteritis. This is one of our major problems in terms of health in Brazil. So these are my thoughts, that we need to reinforce these networks and to build some sort of surveillance system that could deal with older diseases as well.
Many thanks, Fernando. Professor Drexler, you might want to answer as well, and probably you could enlarge briefly on the endemic dialogue that you mentioned. How does it work, and how do other people and organizations, how are they able to participate in this dialogue?
So two things, Mr. Sterz. Let me first reply to the epidemiology, to the scientific question, and I will get to that specific project in the second part of my reply. First of all, I agree 100% with Fernando. Everything he said, I don't think there is a lot of scientists that would disagree. The challenge is to secure the funding, to reserve that funding in the budgets of our governments. That's not an easy task, because we are competing with many other areas, and unfortunately health and within health, infectious diseases have to make a stand compared to many other fields. I think the pandemic should have taught a valuable lesson to our stakeholders, that we are not fighting, so that we're not fighting a lost battle here. We shall see in the near future. I would also like to highlight that epidemiology and and genomic surveillance have to work together. It's completely stupid and pointless to sequence every viral genome from every patient. It will lead us nowhere. The amount of data that is in public databases from many parts of the world is overwhelming. Not all of the regions are equally presented, but from some regions, say richer countries, the States, United Kingdom, Germany, you have so many viruses, so many genomes. Who will keep track of this? This goes beyond the ability of a human individual to actually deal with this and to follow all the updates that come out on a daily basis. So you have to have a smart design. You need epidemiology. The time when I want Fernando to really sequence everything he can is when there is an outbreak and you have an increase in cases. Then I want him to sequence because I need to know if there's a new variant that could be causing this. So we have to talk to each other. And this is another lesson. I didn't want to overload my presentation, but multi and transdisciplinarity are key to this. And especially in the medical sciences, if we cannot even get epidemiologists to talk to molecular biologists, because MDs have a hard time in understanding biologists, and they have a hard time in understanding veterinary doctors, because they are like separate bunkers. And if we don't overcome this, sooner rather than later, we will run into just the same problems that we have run in with COVID. So this is my scientific reply. Well, it became political also, maybe more than I intended. But this is really something that matters a big deal to me. And then for the dialogue of the pandemic, the project is actually unfortunately ending in the end of January. Our funding from your ministry, well, from the Ministry of Foreign Affairs, is ending. And let's see if we can highlight the relevance of engaging in this interdisciplinary and international debate with the coming German government. We are trying to emphasize together with our diplomat colleagues from the German Federal Ministry for Foreign Affairs. I think we all agree on the relevance of this. I have reached out the whole dialogue function through, because we have such a large network in Latin America. We were able to really get people to one table that usually don't talk, which is the lab heads of each individual country. And that's a difficult task, but you need them to talk, because you don't want every country, I don't want every country to try to solve exactly the same problems that a neighboring country has. The virus doesn't stop borders. And if all our nations invest all our human resources and all our funding into developing the same, or at least very similar solutions, we are wasting time and we are wasting money. And we cannot afford this. So the whole idea is talk to each other, work together. And for our government, it means fund transnational research, which is not easily done. How do you tell the government of Brazil to fund research in Argentina and vice versa? And that's not easily done. What we need, the public stakeholders in this discussion, it's really a call for action from my side. We have to fund this, and we have to fund it in a smart way. It's not throwing money out of the window. We have to fund bilateral and multilateral initiatives. And we have to fund this in a way that those people that really have the experience can work with it. So, and that's not an easy task, because who will say what is good research and what is not good research? We are reviewing ourselves. And still, it can be difficult, but there is no other option. We have to engage in this kind of cooperation, or we will fail.
Many thanks, Mr. Spilkey. This is a very important input and a very relevant discussion. I hope we can come back to this point later on. Now, having a look at the schedule, I'd like to come to our next session, of course, which is on vaccines. And I cordially invite Professor Ulrich Lauer and Professor George Elias Khalil-Filyu to our panel. Thank you for joining us. Professor Ulrich Lauer is Deputy Medical Director of the Department of Internal Medicine at the University Hospital of Tübingen, and his scientific focus is on the virotherapy of tumor diseases. This is just showing the multidisciplinary work that we do here. Among others, you, Mr. Lauer, were in charge of the research project Novel Measles Antivirals as the key to the global eradication of measles viruses. Now I believe you are focusing on the eradication of the coronavirus as well, with the research on a new vaccine based on a nasal spray. Mr. Lauer, many vaccines against the coronavirus have already been developed and are in use all over the world. The pharma industry seems to have taken over. Do we really still need further research in the public sector? Please tell us about your research against the coronavirus. I hope Mr. Lauer is already here, joining us. Mr. Lauer, can you hear us? Mr. Lauer, we can't hear you. Your mic is on. Can we already display the presentation? Probably it might be best to continue with our second speaker and come back to Professor Lauer thereafter. So I'd like to introduce to you George Khalil Filho, a professor of clinical immunology and allergy at the Faculty of Medicine of the University of Sao Paulo, and among other positions, he is director of clinical immunology and allergy at the Hospital das Clínicas in Sao Paulo, as well as director of immunology laboratory. Mr. Khalil, currently you are developing various vaccines against Streptococcus, AIDS, dengue, and COVID-19 at advanced scientific stages. Your vaccine against the coronavirus will as well be based on a nasal spray. Please, Mr. Khalil, tell us about your research work.
Thank you, Dr. Sterz. May I share my slides, please? I need a permission.
Yes, you have granted. Thank you.
Thank you. So concerning vaccines, you may know in Brazil that we have a very good national program of immunization. That's the reason why I think we have reached already almost 70% of completely fully immunized people. And we have two producer of vaccines in Brazil that are two government institutions. I think it's too bad that the private sector is not investing in producing vaccines. So we have the Fiocruz and Butantan. I directed Butantan for 6 years. And during that time, we were able to launch the influenza virus plant that when I left 4 years ago was producing something like 60 million doses per year for just the seasonal flu for Brazil. And also I had launched the dengue vaccine that was originally, I mean, the attenuation was done by NIH, but the industrial, all the development and clinical and industrial was performed here in Brazil at Butantan. And when I left, we had already immunized 17,000 people in the placebo-controlled randomized trial. I don't know why it's not ready yet. When these pandemics started, we decided to work in a vaccine, and I'm just going through very quickly to the idea. So the ideal vaccine, we need a cellular and a humoral response. So we need cells, but also we need antibodies. And for that, we have to select the antigen that also they are the targets of immune response. That's not what have been done so far. So far, people took either the inactivated virus or the spike protein that serve as vaccine. And we have to of course, we have to select either an adjuvant or a vector. So when you look at the vaccines that are out there, as I said, they use either a viral vector or RNA, and there are many that are inactivated. So from the beginning we decided to do something different. We are working in a vaccine that's called multi-COVAX, and we started by studying the cells and sera from 220 convalescents, and we have seen in the early beginning that there was a strong neutralization correlates with IgA and IgG against RBD, and for that reason, after trying several antigens, we decided to use the RBD by itself in the dimeric state as the target for neutralizing antibodies. But we need also to have a T-cell response. As you may know, in order to have a T-cell response, you have the different peptides from the virus of the infectious agent have to interact with the HLA-DR molecules in a precise way, because there are some pockets where you have to stay. And so by studying the entire genome or the proteome of all of this virus that has 36 proteins and not just spike, we have selected 66 peptides, and we have tested these 66 peptides. So this is by bioinformatic, and these 66 were promiscuous peptides that would interact with HLA, both class 1 and class 2. So we have those 66 peptides we have synthesized in the laboratory, and then we have tested against these 220 people in order to select the best ones. I'm not going to show, of course, these results. It's just one print of the when we tested in pools, and then we tested individual, and we could then select peptides that would induce a good T-cell responses, both CD4 and CD5, CD4 and CD8, in 99% of all the COVID patients that we have tested, 220, as I said. So from this, we have selected these B-cell epitopes and T-cell epitopes and have designed our antigen. So our antigen is a dimeric RBD that is linked to a strain of peptides that induce a good T-cell response. So this is the antigen, but then we need a formulation because we wanted to do a nasal spray. I'm going to return to that. In order to have a good formulation that would interact with the mucosal cells, for that you need to pass through the cilia, the mucus, in order to be attached to mucosal cells. And for that, after testing more than 50 different formulations, we use now the antigen is binded to a silica nanoparticle, and then this silica particle is surrounded by cyclodextrin in the presence of an adjuvant that's poly-IC, and this is the way that we can actually formulate the vaccine. So the idea is to have intranasal administration, because the mucosal immune response is very straightforward, but also we have a systemic protection. We have we can mimic the natural infection of the virus. We can have a large absorption surface. We have low proteolytic activity, and this area is vascularized and non-invasive. But why doing intranasal administration? It's very simple. The virus enters through the nose. And in these vaccines that are intramuscular, they induce very high titers of IgG, but not very high titers of local IgA. And the IgA is the one that protects the mucosal, and particularly the IgA of the mucosa. So we want to induce the IgA of the mucosa, because we don't only want to avoid disease, but we want to avoid the infection. That's the only way of controlling the pandemics, because otherwise the virus that is in the mucosa can be present even if the person is vaccinated or had already the disease. So in order to do this, if you immunize the mucosa, you protect. There is no infection. If there is no infection, there is no transmission of the disease. That's what we want to do. So by immunizing the mucosa, we induce a secretory IgA, also IgG and IgG in the serum as well, I'll show you, and very high titers of local neutralizing antibodies. But also the resident T cells in the mucosa and the lung will destroy the cells that are infected, and by this way, avoiding completely disease. So when we immunize with our antigen, we obtain very high levels of serum neutralizing antibodies, 10 to the 4th. That's the equivalent of a Pfizer, BioNTech, or Moderna. We induce also anti-seric IgG against RBD after the 3rd dose, as you can see here, very high titers, and neutralizing antibodies after a 3rd dose. And also we had very high levels of serum IgA and against RBD after 3rd dose. And why the 3rd dose? Because, I mean, here in Brazil, almost everybody received 2 doses of the vaccine. So what we need nowadays is to have a booster, and we want to do this booster by nasal immunization, as I told you, to avoid infection, and also because it's very simple. It's very easy to do a nasal immunization. And the other thing that's important as well is that we can put the 4 different variants of concern in 2 constructs, each one with a dimeric RBD. And most importantly, we can detect local IgA after 3rd immunization. That's very high, as you can see here, by looking at the bronchoalveolar lavage, the nasal fluid, and the saliva as well. So the titers are high, and this I'm certain that can avoid the infection. So we are working now in the industrial preparation, and we have already proposed a double-blind randomized placebo-controlled clinical trial for a phase 1 and phase 2 study of this vaccine. So we'll take people that were previously vaccinated with AstraZeneca, that was the most commonly vaccine used in Brazil, and 180 people who received the vaccine, 90 will receive the placebo, and these 180 will be divided in groups that will receive either 1 or 2 doses, and with different amount of the antigen. And then we're going to look at these people and look if there is no infection by examining the saliva of these people we test for antigen. And we are going, of course, going to look at the serum and local immune response in order to show that we can achieve this. So the vaccine features would be to induce neutralizing antibodies and C4 and CD8 responses, induce long memory, no serious adverse events, had broad protection coverage in the population, mucosal and non-injectable administration, preferably single dose, low cost of protection, thermal stability, prevents infection, that's the point, and not only disease, a complete domain of the production technology in Brazil. As you know, the key steps of the production of vaccine, you must go through a vaccine discovery, and for this disease, there are some scientific beads, and they work perfectly. I mean, people use just the spike, but as you can see now, these vaccines that are the 1st generation are not the ones that are going to stay. We need better vaccines. Of course, after the discovery, you have to go through a lot of laboratory standardization, and then the point that is a problem, that is the product development and manufacturing of the first pilot, the first pilot batches. And this is a tremendous possibility of collaboration. I know that in Germany you do it so well, and this is a lack in Brazil. I did develop this a lot at the Butantan Institute when I was there, but there was no continuation of this work, and people that are working on vaccines, and there are 15 projects that have been financed by the Minister of Science and Technology. They are stuck at this level. How we do the bioprocess and how we manufacture in small amounts. Clinical trials, there is no problem. Here in my medical service, we have tried several vaccines, including the phase 2 of dengue and also the phase 3. But this is a gap. Regulatory issues, as you know, Anvisa in Brazil is very good. It's similar to EMA or FDA. It's very serious. So we have all the other areas. So the problem is how we're going to pass this valley of death. And we do need, as my colleague that talked previously, we do need collaboration. And there is a lot to do together in order to achieve our goals. So that's what I wanted to tell you. This work is done with many PIs from my group here at the School of Medicine, University of São Paulo, but also the campus of the University of São Paulo, like Durigon Boscardini. Durigon is going to talk today, and also the pharmaceutical school from Stefano that was involved in all the formulations, and Dani Rosa that was previously our postdoc. We have a team of wonderful people, over 25, that are students, postdocs, and already researchers. From the beginning, we collaborate with Riccardo Gazzinelli that also had received a grant at the same time as mine for developing a vaccine, and we exchange ideas and we exchange reagents. And all this work has been financed by the Minister of Science and Technology and Innovation by this virus network. Well, very quickly, that's what they asked me to do. That's what I want to present, not going into this too much detail. Thank you very much.
Thank you very much to you, Mr. Kali, for this inspiring presentation. And I now would like to invite Mr. Professor Lauer to continue with your presentation. Great to have you back on board. I hope this time the technology is helpful.
Hello, can everybody hear me? Okay, thank you very much. I just have to share my screen. Just a moment.
Do we have the screen as well to share for you?
So I sent over a PDF version.
Probably we
maybe
can.
This can be used.
Yara, can you help out with the presentation?
Yes, you just need to click on the bottom on the right. There's a blue button with a square. Click in there, and you ask you to share your screen. Can you do it?
This one, this button?
After you click, you have to choose the file that you want to share.
To choose the file, you say. I'm a little bit lost. Sorry for that.
Don't we have the presentation to display?
So I sent it over per email, the presentation. I see. Okay, I have this. Okay. I see how it works. I'm sorry for the delay. I'm working on that. Just give me a second. So now it's uploading.
Okay, I might.
So this is.
I might take advantage of this time in asking one question. With all these convincing arguments for nasal sprays as a vaccine, why don't we have more vaccines like this for all respiratory viruses, like the flu, for example? Probably you can touch on this as well.
So indeed, we do have vaccines against flu, which can be applied intranasally. This is a relatively new development, and I'm pretty sure due to the easy application and due to the low costs and the acceptance in the societies, these intranasal applications will go further into the market and become over time a new standard. The problem a little bit about this is that you need specific formulations, and that the knowledge on these formulations is not as expanded as it is with the intramuscular application. But this is on the run, and I'm pretty sure that over time this will gain more importance and more acceptance. So, question, do you see my first slide on the screen?
We see the slide on our session.
Okay.
Okay,
so
here we go.
I'm switching to the second slide and go right into that. So people from the Max Planck Institute of Biochemistry in Munich and from my university hospital in Tübingen, so Professor Fessler, Professor Neubert, Dr. Wiegand, and me, we set up a consortium, and the intention of our consortium is to generate so-called second-generation SARS-CoV-2 vector-based vaccines, which should be truly anti-pandemic by completely blocking virus transmissions. And I just would like to introduce the most relevant questions in the current pandemic. So the most relevant question is, how can we come back to normal lives? We are not back to normal lives yet, and it's not foreseeable when we come back to normal lives. How can we truly defeat the pandemic, and how can we potentially eradicate SARS-CoV-2? So in Germany, we are now battling a massive fourth COVID-19 wave. And this could finally lead to another shutdown and another problems for society and economy and whatever. And as you can see over here, we had a very nice summertime in the midst of the year and are currently really in a dramatic wave we have not faced so far. So the incidence is really growing up very, very fast. And the reason for that is that so far only first-generation vaccines are available, which all are applied only via the intramuscular route of administration. So this is true for BioNTech Pfizer, this is true for Moderna, for AstraZeneca, and for Johnson & Johnson. All are applied only intramuscular. And this leads to a real problem in the way, as you can see over here, this is the German vaccination campaign, and you see about 70% of our people are vaccinated twice. And there is a stagnation right now. So we have to say we have an insufficient vaccination rate. So not enough people could be convinced to get vaccinated. And this is a large problem for the fourth wave. And in 2022, our Robert Koch Institute, which is responsible for the pandemic, already has said it must be assumed that people, despite vaccination, can become PCR positive after contact with SARS-CoV-2, mostly in an asymptomatic state, and then are able to excrete infectious virus despite vaccination. And this risk must be reduced by an ongoing adherence to infection control measures, which means there's no way back to normal living then. And therefore, our experts recommended to continue to observe the generally recommended protective measures, everyday masks, hygiene rules, keeping your distance, ventilations, even after vaccination. So this was in 2020. And this is still true for 2021. And we learned that most of our vaccines, this is the biotech Pfizer vaccine, they allow breakthrough infections. They might reduce the viral load, the duration of the shedding, but still breakthrough infection in vaccinees do occur. This is not only true for the BioNTech Pfizer vaccination, but this is also true for AstraZeneca, for the adenoviral vector-based vaccine. And as you can see over here, in Germany right now we count about 200,000 symptomatic cases of vaccine breakthroughs. This means 3 out of every 1,000 fully vaccinated people have been shown to have contracted the disease while also showing COVID-19 symptoms. And we have to take into account the many, many asymptomatic vaccinees who exhibit vaccine breakthroughs, who could be performed as super spreaders, not knowing that they are really spreading the virus. And all these components build up that Germany currently sinks into dark colors. Dark colors mean high incidence. You see over here, this is mostly in the southern parts of Germany right now and in the eastern parts, but it's foreseeable that also the other parts in the north and in the west of Germany will get at the same stage quite quickly. And the reason for that is too much unvaccinated people, in addition to breakthrough cases from symptomatic and mostly asymptomatic cases. So there's a campaign now going on for booster vaccinations, but so far we only have nearly 5% of booster vaccinees. This will take time, and the wave is already on the run. And the most problematic thing, as we heard before by Professor Khalil, is that the infection with SARS-CoV-2 takes place in the upper respiratory tract. The replication of the virus is strong over here, and it decreases when we go in a direction of the lower respiratory tract, as shown in a recent Cell paper. If we look at natural infection, it's a respiratory virus. So infection would lead to the antiviral immune response also in the respiratory tract. Then when we look on the first generation vaccines, which only can apply today, up to today, via intramuscular injection, this is a quite non-physiological route of vaccine application. And this leads to a major disadvantage for full protection against respiratory viruses, because the airways are not enough protected by these first generation vaccines. So you see it over here, most part of the respiratory tract are unprotected by BioNTech, Pfizer, Moderna, AstraZeneca, Johnson & Johnson, and most other developments which are applied intramuscularly. And this leads to an unstopped virus transmission, to waves and waves of SARS-CoV-2 transmission in this pandemic. In contrast, when we will apply second generation vaccines intranasally, we will achieve protection also of the airways. And this could help to completely block any virus transmissions, to be truly anti-pandemic, and to bring down the current wave and to inhibit or dissolve other waves. So these second generation vaccines, as we heard already, are easy to apply. This will help in terms of acceptance. This will help in easiness of application. And as you can see over here, non-protected lungs are easily infected by these little viruses, leading to the destruction of the airways and death in oxygen need. But when we have protected our lungs via intranasal vaccination, and we could induce this Y-shaped immunoglobulin A antibodies, these antibodies will really take away the viruses which income, will neutralize them, and help to protect our airways and help to protect our lives. So this kind of antiviral immune response already in the first line of defense in our respiratory tracts is still needed and presumably can only be done via intranasal application of second-generation vaccines. So we are working on a sender virus, vector-based vector, which is listed on the WHO list of preclinical candidates so far. That means that we plan to enter clinical trials end of next year. And as you can see over here, if we would summarize the advantages of intranasal application of a vector vaccine, this is prevention of initial infection by SARS-CoV-2 of airway cells in the respiratory tract by secreted IgA antibodies as a very important first-line immune response. Infections then can be stopped very early before the virus can spread in the body. And the induction of the mucosal antibody response against SARS-CoV-2 is followed by a broad and lasting specific cellular and humoral immune response. So also the organs and the lymph nodes and whatever in the body are protected via this application route. And genetically encoded safety features of our vector-based approach will qualify the application also for the broad population, for children, and also for the elderly. A special innovation could be that based on the sender virus backbone, which will be genetically manipulated to encode the spike SARS-CoV-2 gene, we could build particles which on the surface, vaccine particles which on the surface only will exhibit spike proteins. So for our immune system, this would not lead to an anti-vaccine immune response, which would neutralize such a vaccine particle. Instead, first generation vector-based vaccine, like the one from AstraZeneca, which is based on an adenovirus, still is an adenovirus on the outside. And we do know reapplication is required in this pandemic. So if you come a second time, maybe a third time with this foreign virus, this might be neutralized by anti-adenoviral antibodies. And so efficiency over time could decrease due to vector neutralization. So the advantages of our second generation COVID-19 vaccine is to induce a profound mucosal immunity. This should enable first-time sterilizing immunity, and this should build up the chance for a worldwide eradication of SARS-CoV-2 due to the stopping of any further transmissions. I just would like to summarize, pandemia, from our point of view, will not be overcome by the current first-generation vaccines. We are working on the development of second-generation vector-based vaccines. We already have expertise from the generation of vaccines against another respiratory virus, the respiratory syncytial virus, RSV. This is a blueprint for our developmental approach. We have established structures for fast implementation and translation, a vast knowledge pool for study documents, study process, and we have a very nice team working very hard on that. Thank you very much for your attention.
Many thanks to you, Professor Lauer, for your inspiring and impressive presentation, clearly showing the necessity to continue with research on vaccines. Now the floor is open for the audience to ask questions, and we already have one first question in the chat, which is, what is the sample size used in your clinical trial? I guess this is a question to Professor Kali.
Okay. Well, first of all, thank you. I mean, I didn't know that Dr. Lauer was going to present also an intranasal vaccine. I think it's fantastic. I mean, it's very synergic. I think we have convinced the audience that that's the way to go. So the proposal for this phase 1, phase 2 is just 270 people. 90 would receive placebo, and 180 would receive the vaccine in different ways. I mean, different dosage and 1 or 2 installations. That's for phase 1 and 2. For phase 3, we have to see at what the point of the pandemics will be to have a good structure of the phase 3. Because if you want to show that we can avoid infection and the virus is not circulating in Brazil, it may be a nightmare. But if the virus is circulating, then it would be easy to show. So I think that I cannot do a program the phase 3 till I have I have to do it according to the pandemic stage. I mean, in Brazil, the pandemic is fading like it did in Europe. We don't know if it's going up again by the end of, because we are entering summer. So it's exactly what's happened in Europe. So I hope that in falls, we don't have the same peaks you're having, but we don't know. So we don't know because the Delta is there, but the pandemic is going down. Even with Delta, we could identify that almost all the cases in São Paulo are Delta now, but it's not causing the same problem in Europe. I don't know if it's coming or not. So phase 3, I don't know. Phase 1 and 2, these are the numbers.
So I would comment on that the best time to come over to Germany and Austria is now to do clinical trials. Yeah, yes. We have the numbers, we have the cases, so this just turned totally around. Yeah, so come to Germany.
Let's work together, Dr. Lauer.
Very nice place for clinical studies.
Well,
we should link it back. I mean, if the pandemic is worse in Brazil, you test your vaccine. I can help you test in Brazil, right. and right. the other way around.
These are great, great final remarks on this session. Thank you very, very much. I believe that this pandemic can and should be a booster to innovation in the research on vaccines, and I hope it will be in the future too. Now I'd like to come to our third and final panel, because we are already lagging behind a bit on the time schedule, and we will dare to take a look at what might be expecting us in the future. And I cordially invite to our session on epidemiological surveillance of emerging pathogens in wild animals, Professor Edison Luis Durigon, Professor Dr. Marcia Goethert, and Professor Stefan Laufer. Great to have all of you with us. Your contribution is very important to us. Starting with Professor Edison Luis Durigon, he is a full professor of virology at the University of Sao Paulo. He was the representative of South America in the Biosafety Advisory Group of the World Health Organization in the year 2004. And Mr. Durigon, currently your research encompasses clinical and molecular diagnosis, human and animal viruses, and many other kinds of viruses, are there asymptomatic, respiratory, wild, or emerging. I guess we will learn a lot from you about viral genetic diversity and emerging viruses. Mr. Durigon, the floor is yours.
Thank you very much, Christian. I'd like to thank all the Brazilian authorities for my invitation, especially for the German authorities that is turning this possible to talk about this virus in the wild animals reservoir. That is very exciting. And it's a very, very big pleasure for me to talk in this seminar, special for the German side that the University of São Paulo has for a long time, a long and strong collaboration with the German. I remember clear in 2003 when the first SARS epidemic starting and transforming the pandemic in the North Hemisphere. We get a very, very opportunity collaboration with Dr. Manfred Weyling and Frankie Hofer from Freiburg University, that we get the primers and the controls and make the first real-time PCR for SARS-1 in Brazil. It's make us to make a laboratory chain and try to do surveillance in this virus. And you get 2 suspicious cases that come to São Paulo in Ribeirão Preto City. And that is our first nice collaboration. And after that, after 16 years, I never imagined that we are facing the SARS-CoV-2 pandemic. And we, again, have a very, very strong collaboration from Charité University, from Dr. Christian Drost and Victor Carmen that share their protocols with us. Dr. Dresler is a very good friend of us. I have 2 students, 2 postdocs in his lab right now. And they articulate all the way to send for us the primers, protocols, and the synthetic RNA for Brazilian scientists to start to do the real-time PCR for SARS-CoV-2. It is before the pandemic in Brazil. It is middle of January 2020. That is the reason that I am very pleased to be here. I would like to put in my presentation. Let's see if I can share. Okay, I can do. Let's see. Everybody see my presentation? Not.
We do see your screen, but not the presentation.
Okay. Let's see if I can do something better. Can you see my screen now?
The screen and the presentation. Now, here we go.
Yes, okay. I cannot see anyone because I only have the presentation, but I hope it's clear for everybody. Okay, I will talk about our national network of epidemiological survey of coronavirus SARS-CoV-2 and the other emerging pathogens in bats, birds, and the other forest reservoir in the context of One Health. As everybody knows, Brazil is large biodiversity in the world. And this project that we call it PREVIR is having strong support from the Minister of Science and Technology and Innovation and the CNPq. And the coordination is Dr. Clarice Arns from University of Campinas, Dr. Helena Lage from University of São Paulo, Campus Pirassununga, and by me, Edson Durigon from University of São Paulo, Campus São Paulo. Just for situate everybody in this presentation, give some background that we are doing for all these years. That's the screen here is from São Paulo City. São Paulo City is the largest city in Brazil. We have almost 20 million people if we count the surrounding São Paulo. And the University of São Paulo, this is our department. This is a microbiology department at University of São Paulo. It's one of the largest universities in Brazil. And we do the virology here. We building a BSL-3 facility, and we're building a field facility to try to looking for different emerging viruses in different reservoirs in Brazil. This laboratory started in 2001 with West Nile virus disease when the pandemic started in the United States. It's endemic because they never changed the continent. And then in 2003, SARS-1 is starting, and 2005, we face it with all this infrastructure, the avian influenza virus, high pathogenic strains, that was the possibility to come to Brazil and give a big bang the pandemic in the world. That is the virus that until now we are more concerned about. In 2009, we again faced the pandemic from influenza virus H1N1 that's come from Mexico and spread for the whole world. In 2015, we prepare straight with Zika virus, and we have a strong collaboration from Dr. Dressler from Charité University to do all the diagnosis again. And this Zika virus transformed in a pandemic. And then we have several epidemic in São Paulo. We have measles epidemic in São Paulo. We have yellow fever epidemic in São Paulo. And then in 2020, we face again with the SARS epidemic that's coming, the COVID pandemic in the whole world. Historically, just to put it, our first expedition looking for wild animals and their reservoir for emerging virus that has potential for pandemic, starting in 2005. Our first expedition was for the Amazonas area. We got the Manaus state and the Belém state. We have these two cars that we just drove from São Paulo City to Western Amazonas in Porto Velho, Rondônia. And then we drive for almost 1 week to Manaus, and then we take a boat with this instructor and go to Belém, make all these captures and back to São Paulo again. This is our great and first experience for looking for different viruses. And then some results that we have in different expedition. This is in the Amazonas area. That we're looking for influenza virus in migratory birds that came from the North Hemisphere to South Hemisphere. Every year we have around 5,000 billion birds that immigration from the North Hemisphere to the South Hemisphere. They come in around November, stay until April, and May they fly back to North Hemisphere. And this is the way that we do these expeditions in the Amazonas area. It's not easy to drive in this area, and then the boat is the major attitude. That's our liquid in nitrogen, and this is our team for looking for birds. This is Canela Island. I would like to introduce you. It's a very nice island in the Amazonas region. That is, we collect the birds there, take out the samples, put it in liquid in nitrogen, and bring back to São Paulo to do all the work. And this first expedition, we get some exciting results with influenza virus H11N9. That's our first isolation in Brazil. And it was not surprising, but we are seeing that this virus, it's happening in the same year, very close, in New Jersey Bay, in Delaware Bay, and you have the same 1 year ago happened. And this is, we think that this virus can migrate every year from the North Hemisphere to the South Hemisphere and change different reservoirs. This is from the Northeast region. That's not, we started the Amazonas back to the Northeast region. This is in 2009. We focus in Coroa do Avião Highland. It's like a paradise for the shorebirds. They migrate from the North Hemisphere and stop here for breeding. In the Northeast, we use, because there's so many shorebirds there, we used to collect the feces and bring it to São Paulo to do the influenza. We look for the bands, to classify by photographs the same species of birds. We collected the figs there and bring to the diagnosis. We have opportunity to track the dates and the location for one shorebird from the northeast, the Amazon, the Maranhão area. It's the northeast of Brazil, that they migrate back to the north hemisphere, and then this was in February, and then they back in August, in September again, to the same place that they live from Brazil. Okay, that is take the virus there and bring the virus there, and that's another hotspot that we have in Brazil. We are in the north of Brazil, northeast. Now we are in the extreme south of Brazil, is Lagoa do Peixe. It's a very hot spot for avian influenza virus in wild birds. And we find here different lineages in South America. We are very surprised with all this amount. We get the H6N1, we get the H12N5, H9N2, and we also have H2N2 that is caused the second pandemic of influenza virus in human in the world after the Spanish flu, and disappear for complete and give place for H3N1. And now we found this same virus in one Calidris fusiculus. It's a shorebird in Lagoa do Peixe in 2012. Okay? But we look for this H6N1, that is this virus that is very singular isolation, and we found that H6N1, it's a South American lineage if you compare with a different lineage. We also did in the same time bats looking for exotic disease, because we didn't have this disease. We didn't, because now we have several surprises in Brazil for Nipah virus, other paramyxovirus virus, and coronavirus in bats. That is our way to work in the field. We do the glove box, collect the bats, put it here, and take some bloods and everything. And we did a lot of bat coronavirus in Brazil. I have my student is that Luis Gustavo Góes, that was my postdoc, and then now he's working with Dr. Dresser in Charité. And Gustavo did a nice work in Brazil looking for coronavirus. We get coronavirus from the undisturbed forest area, like Foz do Iguaçu National Park and the Amazonas Forest. In the fragmented areas, like Central Park in the states of São Paulo, it's Parque Ibirapuera, and some urban areas in Northeast and the southeast of Brazil, and we find almost 26 different coronavirus from 10 different bat species. We have 11, we'll have 24 alpha coronavirus and 2 beta coronavirus in these bats, with 3 potential new coronavirus. We did all the phylogenetic and have a lot of work in collaboration with the German too, in this case. Now, in February 2020, we started the project with the Minister of Science and Technology, Rede Vírus, we call it Previr, okay? Because we had some cases in Brazil that make us to looking for new virus surveillance in Brazil. We're scared not only for coronavirus that in that time is almost coming, but for some other virus that we already had in Brazil. We had some arenavirus epidemic in Brazil. We have the first case of arenavirus, they call Sabiá virus, in 1990. And then we have in 2019, one case in São Paulo State, in a cave. It's a man that visited a national park in São Paulo, Brazil, and get this nanovirus and die for that. It is a BSL-4 facility, and Brazil doesn't have any BSL-4 facilities right now, but we will do. In February 2020, we get the first 2 confirmed cases of COVID-19 coronavirus in Brazil that we are isolating our laboratory and put a lot of effort to distribute the virus for the whole country. And in December 2020, we get a 4-year-old girl infected with influenza virus from avian resource, this H1N2 virus. That's fortunately, this is a very mild disease, but we are scared for the transmission for human to human. And also, we still have some few cases of rabies virus that came from bats and fox in the northeast of Brazil. In September this year, we have a sad case of a toddler that died of infection for rabies virus in northeast Brazil, state of Maranhão. Okay. Then the Preavir, that is the project that we are talking here about, it makes sense for us. And we put all our effort to building a team, to looking for different regions in Brazil, and put all the network that we can collect samples from wild animals, do all the data collection, make swabs from these animals, collect blood and tissue, do the PCR screen, sequence, do all the phylogenetic analysis, and then we also can do some in vitro experiments in this virus. Okay. The eco-surveillance of this virus in wild animals from Brazilian domains, we get the Amazonas area, Atlantic Forest, Cerrado, Pantanal, and wetlands, where we are. We are here, that's Brazil. We are in the Amazonas forest. We have a team there that screening virus from 3 and 4 different locations in the Amazonas forest. We have our wetlands, this Pantanal, it's in the center of Brazil, that we have a very nice team there that collect bloods in different points in the Pantanal areas, that is the Poconé area. We have a team in the northeast of Brazil that has a remaining Atlantic forest space, collecting birds and bats. And we have a team in the extreme south of Brazil, Atlantic forest, Santa Catarina, that is collect samples from these animals. What we have until now, we have 2,353 bat samples and 2,562 bird samples from November 2020 until October 2021. That is the different biomes that we have. We have many cases from Amazonas, many cases from Atlantic Forest, Pantanal, and the Cerrado. That's all part of our Preavir project. That is the most common species that we found from the bats. That's Stornira, Artibeus, Epitacus diminutus, Myotis, Glossophaga, and Carolia. That's some bird species that we collect in all these teams from the Amazonas areas to the extreme south of Brazil. We have a lot of Sterna oriundo, Calidus fusiculus. Remember that this Calidus fusiculus was the shorebird that we detect the major numbers of influenza virus H6N1. And what we get from coronavirus, we have the majority that we have until now is alpha coronavirus. We detect the alpha coronavirus in 46 samples from 7 bats collected in Atlantic Forest. We have the majority of these samples coming from northeast of Brazil, is the Severino group, that we have these numbers: 7 in Sturnira, 10 samples isolates from Carolia, 1 Artibeus, 16 for Philostomus, 1 for Desmodus. And we have from the southeast of Brazil 4 samples detected, 1 from Diphyla, it's Haematophagus, and 3 from Sturnira. And in Santa Catarina, that's our extreme south of Brazil, we have 6 different coronavirus detected. Okay. That is the number of samples that we tested, and we detected in the total 46 samples, 46 alpha coronavirus from more than 500 coronavirus screened. That's something very interesting. For example, from the Diphyla, that is the Haematophagus, we detected the coronavirus, alpha coronavirus in swabs from oral and the rectal swabs, but we also detect in different tissues. But we didn't have this in this Sturnira region. It's other species of the bats. We have some suspicions that some species of bats spread differently the coronavirus than others. That's maybe make some differences in the reservoirs. Another results that we are looking for and think about, because we have the high identity of coronavirus sequence from the same bat species within a bit distant geographical location. We had more than 3,000 kilometers between different samplings, and we get a very, very, very high identity when we sequence the spike region. That is, we know that these bats do not get a migration from the north to the south of Brazil. Then we are thinking about each species has their own coronavirus, and maybe this is make a difference. From the birds, in these times we test samples from almost 400 birds, but all still now is negative for coronavirus, avian influenza virus, and Newcastle disease virus. I would like to thank all the people that collaborate with us, especially for the financial support that came from Ciência technology. And we hope that God is to continue to be Brazilian and protect us for the next pandemic. Thank you very much.
Many thanks to you, Mr. Durigon, for your input on your valuable research work. These findings are core for our topic of pandemic preparedness. In fact, it shows that we are far from being safe from new pandemics. Now I will pass over to, in fact, 2 speakers for our next presentation. And so I can not only introduce 2 experts, but we will also learn about an ongoing German-Brazilian research cooperation. So I'd like to introduce to you Professor Dr. Marcia Göttert, who is a pharmacist and a permanent professor of the graduate program in biotechnology and the graduate program in medical sciences, as well as a visiting professor at the Department of Medicinal Chemistry at the University of Tübingen in Germany. Your research is, among others, dedicated to the development and validation of bioassays for quantitative determination and identification of small molecules with anti-inflammatory and anti-tumor activity, as well as the identification and characterization of the cellular and molecular processes involved in order to develop new medicinal or biotechnology products. And you team up with Professor Stefan Laufer, who is chairman for pharmaceutical chemistry and head of medicinal chemistry at the Department of Pharmacy and Biochemistry at the Eberhard Karls University in Tübingen. And your main research interests are inflammation, autoimmunity, and cancer. In addition to your academic work, which is encompassing the authorship of over 400 publications and 15 book chapters, you have been quite busy in business as well. Former positions include the head of research and development and the membership of the management of Merkel GmbH, and later of the supervisory board of BioPro Baden-Württemberg GmbH. You are co-founder of HEPA Reganix GmbH and Cair Biosciences GmbH, and you hold over 40 patent families with over 300 national applications. Great to have both of you joining us. What are your findings on the coronavirus and how can we prepare for future pandemics? Many thanks.
So first of all, thanks a lot for the invitation and the possibility to present here. As we are almost an hour behind schedule, I'll try to speed up my presentation. As already said, we are located here in Tübingen University. Tübingen is, as you may see here, close to Stuttgart. All of you may know Hamburg, Düsseldorf, Frankfurt, Munich, Berlin, the big cities. And Tübingen is in the very southwest. We are one of the 11 universities of excellence, and we hosted a couple of Nobel laureates, a few of the last ones in our field, life science chemistry, you may see here. The country of Baden-Württemberg is a very rich state in education. We share many, many universities, Max Planck Institutes, and so on. And what is even more important, we are hosting the Brazilian Centrum of the state of Baden-Württemberg. And our job is to make the bridge between Brazil and the southwest country of Germany, Baden-Württemberg. And what we will talk today is exactly such a project where we bridged Baden-Württemberg to Rio Grande do Sul, and in Rio Grande do Sul, especially Lajeado, and in Lajeado, at this place, very famous university at Univates. So what you see here is now bridging Tübingen, a very old traditional university founded in 1477, founded even before Brazil was discovered, and Univates, a modern new university in the very south of Brazil. We founded in Tübingen almost 10 years ago the so-called Tübingen Center for Academic Drug Discovery. And what we do there is making from an academic platforms drug discoveries with the aim to make translational research, bringing inventions from bench to bedside to first into man. And this Tübingen Center for Academic Drug Discovery is a platform in the Excellence Initiative of our university. As part of this center, we have established the largest proprietary kinase inhibitor collection currently available in the world, with more than 10,000 proprietary compounds, all made in academia in our group. And this is in an automatic storage system, as you see here. And these compounds are annotated with 1.3 million biological data points, fully annotated in a software system, NIME. And out of this system, we started 2 years ago, 1.5 years ago, to work not only on cancer and inflammation, which was our initial field of interest over the last couple of years, but also to explore this database for new findings for coronavirus. And we found in this, or we have worked in this field on P38 MAP kinase inhibitors, which were initially developed for rheumatoid arthritis, which are now switching over to cancer, and now very, very, very latest to SARS-CoV-2 virus as well. And this work will be later presented by Professor Marcia Göttert. We are quite active in this field. Only 1.5 years after we started, we have already published 12 highly cited papers in very reputed journals. You see here 2 times Nature Communications, Angewandte Chemie, Scientific Reports, and so on. So we were quite successful in exploring this new field based on our Tübingen Center of Academic Drug Discovery. And now I'm very, very happy to hand over to Professor Marcia Göttert, who initially did her PhD in our group here, then she went back to Brazil, is professor at the Univates, and is currently guest professor at my institute here.
So everyone hears me well? Okay. Also, thank you for the invitation and the opportunity to participate in this event today. And thank you, Professor Lauer, for your nice introduction. I'm a Brazilian, a pharmacy, and I have collaborated more than 50 years with a group of Professor Stefan Lauer. And our collaboration has been intensified in 2020, precisely with the pandemic COVID-19. Today, we will speak a little bit about our collaboration and more exactly show a little bit about our results and our main topics in which our collaboration is focusing. The SARS-CoV-2 from genomics to drug design and coronavirus SARS-CoV-2 detection and assay development. The SARS-CoV-2 from genomics to drug design try to understand how the SARS-CoV virus and SARS-CoV strains present in Brazil evolved and how they behave, and so to find strategic targets to look for candidate molecules. And another topic is about the detection and the assay development. Because of our years of collaboration, we have consolidated know-how and developed tests for detection of these new molecules. And so this allows us to know how molecules behave together with the protein of interest. Sorry. So this is our first published article, our first results based on the first steps of our project, as you can see, focus on genomic annotation, identification of mutation rates, and in these genomes, and finally evolutionary analysis. This then allowed to the construction of three-dimensional models of the virus protein, and finally the search for the most stable compounds, and I will show you this in the next slides, and the most active at the active sites of interest. And this first work we published in 2021. We evaluated 627 genomes from the Brazil SARS-CoV-2, and we identified 109 mutations. And these mutations, some of these mutations were later confirmed in some studies performed in Brazil. So we know that the mutations play a very important role when it comes to new infections, reinfections. It's important to search new molecules, how these molecules can interact after a protein get this mutation, and how each alteration in the structure and the active site of can interfere in the therapeutic effect. So this also in the same work, we showed the most prevalent mutations observed in 4 viral proteins: spike, nucleocapsid, RF3, and NC. And these proteins are involved in different stages of the viral life cycle, and especially in the interaction with the host cells. These proteins are potent antagonists of the interferon system, which is very important for our natural immune defense. And on the other hand, in response to this infection, we have the activity of mitogen-activated protein kinases, and especially the P38 pathway, which play a very important role in inflammation. Activated some transcription factors and leading to the production of inflammatory cytokines. So, and beside this, the P38 MAP kinase is associated with lung and cardiac lesions associated in the COVID. So leading to the cytokine storm. And why we have this relation with the P38? Which relation we have? The SARS-CoV virus, when infect the cells or and other respiratory RNA virus, can upregulate the activity of the P38 MAP kinase directly. This perhaps for the replication purposes. And this unique viral mechanisms of the SARS-CoV via ACA2 disable an important mechanism that regulate the P38 MAP kinase pathway, controlling vasodilatation, anti-inflammation activities, and others. And this combination leads to uncontrolled P38 MAP kinase inactivity, where we have vasoconstriction, oxidative stress, and increased vascular permeability. And additionally to this, the loss of the activity of the AC2 when the virus binds to the receptor allow angiotensin II mediated activation of the P38 to predominate in the lung and in the heart. And this positive feedback leads to an increase in inflammatory cytokines production and with a strong response and hyperinflammation. And these cytokines production exhibit a great individual variability in their function, and also different target cells play a different role. And what we know is that in COVID, some cytokines play really important role in severity and maybe are associated with worse prognosis of the patient. And some examples that you can see here in this table is interleukin 1, 6, 10, the macrophage inhibitor protein 1 alpha, 10F alpha, were really involved in a better prognosis of these patients. And amount of these, the interleukin 6 has been shown to be a marker, as you can see here in this graphic, for mild and severe prognosis of COVID patients. So all these informations and that science and different studies show to us not only get a benefit in the development of vaccines, but also the knowledge about the behavior of the virus, different proteins that are involved in this response, and which pathways are activated. And the P38 is an important role, plays an important role in these mechanisms, especially when conducting to a hyperinflammation state. And this is for us interesting target to investigate. So with this in mind, we could suggest a dual inhibitor which prevents replication and also that could have anti-inflammatory activity or which maybe attenuate it. So with this background, we sought to evaluate different MAP kinase and special P38 MAP kinase inhibitors against the potential to interact between the RDB in the AAC2 interface. And we used different PDB codes to evaluate the different compounds that we were analyzed, and one specific compound that were presented by Professor Lauer, had really a good interactions by this bioinformatic analysis in this interface by binding by the RDB. So in order to confirm this, to reinforce this result, we worked in the development of a specific assay with RDB and the spike trimer to detect how these inhibitors bind, and if these inhibitors can inhibit this interaction between the RDB or spike to the AC2. And what we see here is that this compound in very low concentrations by 1 nanomolar inhibit more than 50% of this interaction. And when we use the full spike, the trimer protein, this interaction is reduced significantly. So after this, as this is a P38 MAP kinase inhibitor, and we know that this compound inhibits cytokines release, we investigated in our monocyte-induced macrophage model the real release of TNF-alpha after 3 and 24 hours, and what we see is that the compound inhibits the release of TNF-alpha, but this release was decreased after 24 hours. Okay, but what we know is that in COVID-19 we don't have just one single cytokine that is circulating, just one cytokine, but we have our own controlled relief of several cytokines, and as I showed you before, interleukin 10, 6, and TNF-alpha are good markers and are high in the when we got more chronicity in the disease. So we established a new model with macrophages and induced a strong cytokine release with a very high concentration of lipopolysaccharide during 24 hours. And after this, we added our compound to see if this compound can reverse the release of the cytokines. And surprisingly, we can see that here at 40 hours later, we have a significant reduction, also again here by TNF-alpha, interleukin 6, and interleukin 10. So, also this was tested in an ex vivo model, widely used to identify inflammatory molecules. It's a human whole blood model. and important way to search new molecules, since we can consider important factors such as the plasma binding, drug solubility, toxicity, and cell permeability in this assay. And also we evaluated TR70 cytokines, and the most important cytokines were reduced, and additionally the macrophage-induced protein 1 alpha. Finally, supporting our idea of a dual inhibitor and based on the bioinformatic interactions with the RDB, we evaluated this compound in various cells and also in a reinfection model with CALU and CACU2 cells. And in these models, the activity remains around 5 micromole. And last and therefore not least, let me show a little bit about another work and about the assay development. It is not enough that we have good treatments if we don't have fast, sensitive, and mainly low-cost detection methods to apply these treatments. Today, as we show in other presentations, we have PCR lateral flow tests, which are very sensitive, but of significant cost, especially in developing countries like Brazil and Africa. And based on this and thinking and alternatives to detect the COVID-19, we are working along other essays on another assay in a very simple, fast, and low cost. This work was in partnership with the MNE, with our work with nanobodies produced in alpacas, are very specific and with a low IC50 range. And it is very, very high affinity for the ADB, compete with the ligand sand, and we use this in our model to gain specificity and sensitivity in our assay. And this is our first and the final model that we have adapted on a simple swab. In our first steps, we validated the STEM system with a spike recombinant protein, and we can detect this in between 5 and 10 minutes. After the validation, we evaluated this with the virus and compared this with a standard lateral flow test that is on the mark. And we have a good surprise here that our sensitivity was 4 times lower than the standard test on the market. And at last, in the last months, we published our last work together, a beautiful review about COVID-19 and neuropsychiatric disorders and COVID, a very important theme that should be more discussed, more explored, and yes, due to the lack of information that we have. So I thank you so much for your attention, and a special thanks for all our collaborators and our group that are working together with us. Thank you so much.
Many thanks for your contribution, both of you, and I believe testing will be a topic we will have to follow furthermore. Now the audience is open for questions. Does anybody want to step in? If that is not the case, we have already run out of time, so I might pass over for the closing remarks. And I cordially invite Secretary Morales to deliver the closing remarks. Many thanks for joining us again, hereby underlining the importance of today's webinar. The floor is yours.
Christian, thank you so much, and I really appreciate the opportunity to talk, especially with the great scientists that we have. I would like to thank mainly the scientists, the Spilk, Dexler, Lauer, Dr. Khalil, Durigon, Marcia, and Stefan for these great presentations that shows particularly the potential of the interaction that we can have between our countries facing the pandemics. I could stress the possibility in genomic, the observation of animals in the wild, but also in the synergies in vaccines. We saw here presented by Dr. Khalil and Dr. Lauer the importance of research vaccines such as the nasal possibility that has a lot of advances, and we could work together. So thank you so much, and this is the beginning of this interaction. We have so many other ones, but this is one more, and I thank you so much for this opportunity. Congratulations to all the scientists in the collaboration that we can do together between our countries. Thank you.
Thank you very much, Mr. Morales, for once again stressing the importance of our bilateral cooperation. The aim of this event clearly is to give an impulse for more cooperation in research and development in the field of health, and especially pandemic preparedness. We hope to have inspired the participants as well as a broader audience with new aspects, ideas, and incentives for new exchange and cooperation. Of course, all organizing bodies of today's webinar are happy to be of help in this respect. Thank you very much for your participation, with special thanks to our speakers, and please stay tuned for upcoming next webinars. All the best to you. Goodbye.
Thank you.